Dear colleagues, my name is Anastasia Ilina. I am a research assistant of the Laboratory of Molecular Mechanisms of Aging at the St. Petersburg Institute of Bioregulation and Gerontology. I am a PhD student of the Department of General Pathology and Pathological Physiology at the St. Petersburg Institute of Experimental Medicine. Today I'd like to focus on potential targets for the neuroprotective effect of short peptide in Alzheimer's disease. It should be reminded that Alzheimer's disease is the most common neurodegenerative disorder that causes progressive memory loss in older people. Currently, several key pathogenetic links of AD are identified, including protein misfolding and aggregation, neuroinflammation, synaptic dysfunction, impaired neurogenesis, blood-brain barrier breakdown, oxidative stress, mitochondrial dysfunction, and circadian dysfunction. Let's talk about these links. The main histopathological features of AD are amyloid plaques and neurofibrillary tangles, which are formed as a result of misfolding of the amyloid beta peptide and tau protein. Amyloid beta consists of 38 to 43 amino acid residues and is formed by sequential cleavage of transmembrane amyloid precursor protein by beta and gamma secretases. Tau protein stimulates the assembly of tubulin into microtubules in the brain. The biological activity of the tau protein is regulated by the state of its phosphorylation. Abnormal hyperphosphorylation of tau protein leads to the formation of neurofibrillary tangles, causing a cascade of pathological disorders in the functioning of neuron in AD. Neuroinflammation generally refers to an inflammatory response within the central nervous system. The process is marked by the production of pro-inflammatory cytokines, chemokines, prostaglandins, reactive oxygen species, and other small molecule messengers by innate immune cells in CNS. Synapses are the basic units that transduce information among neurons. Synapses are formed when navigating axons identify their correct target cells. This is followed by specialization of presynaptic axon terminals, which release neurotransmitters, and the maturation of postsynaptic compartments, which transduce the incoming signals by neurotransmitter receptors. The number, structure, and function of synapses change over time. This process, termed synaptic plasticity, underlies the cognition. Aberrant function of synapses, termed synaptic dysfunction, is most closely correlated with cognitive decline in AD patients. Upon postmortem examination, the brain of AD patients have significantly fewer synapses than those of healthy controls. Neurogenesis is the process by which new neurons are formed from neural stem cells and incorporate in neural circuits. Traditionally, neurogenesis was thought to take place in the mammalian brain during embryogenesis. However, numerous studies in the last 3 decades provided substantial evidence suggesting the formation of new neurons in specific brain areas, including the dentate gyrus of the hippocampus and subventricular zone of the lateral ventricle in rodents. Neural stem cells pass through different developmental stages before getting incorporated into the hippocampus circuits and becoming mature neurons. Increasing evidence suggests that new neurons play a crucial role in learning and memory. In turn, a wide range of evidence suggests that neurogenesis is impaired in Alzheimer's disease, contributing to memory deficits. The blood-brain barrier is formed by tightly sealed monolayer of brain endothelial cells, which keeps neurotoxic plasma-derived components, leukocytes, and pathogens out of the central nervous system. The capillary length in human brain is 650 kilometers, which accounts for greater than 85% of total cerebral blood vessels length, providing the largest endothelial surface area for solute transport exchange between blood and brain. The BBB controls the composition of neural internal environment, which is essential for neuronal and synaptic functioning. BBB breakdown has been shown in Alzheimer's disease and is associated with cerebral blood flow reduction and impaired hemodynamic response. Recent studies suggest early BBB breakdown and vascular dysfunction in AD before cognitive decline. Circadian rhythms, defined as oscillations with a period of 24 hours, are fundamental components of mammalian physiology and serve to coordinate physiology with external signals, such as light-dark cycle. The oscillations of the sleep-wake cycle are one of many systemic circadian processes in the organism that are controlled by suprachiasmatic nucleus of the hypothalamus. Circadian rhythms are maintained by the activity of the transcription factors, which control the transcription of many genes, including their own negative feedback repressors. Dysregulation of circadian rhythms are common symptoms of dementia in AD. Mitochondria play a crucial role in bioenergetics and respiratory functions, which are essential for cell viability. Mitochondrial morphology changed rapidly in response to external factors and changed in metabolic status. Over the last few years, remarkable efforts have been made to investigate the impact of various forms of mitochondrial dysfunction on the pathogenesis of Alzheimer's disease, such as excessive reactive oxygen species production, mitochondrial dihomeostasis, loss of ATP, defect of mitochondrial dynamics and transport, and mitophagy. It has been established the crucial role of alteration in mitochondrial function and oxidative stress in the pathogenesis in AD. We carried out a study focused on identifying the potential targets for the short peptides in the pathogenesis of AD. First of all, the binding sites of peptides with DNA were identified by molecular modeling method. On the next step, these sites were found in more than 200 promoters of genes involved in the development of pathogenetic parts of AD by using the eukaryotic promoter database and Postgraduate. Moving on, the cluster analysis of the functional relationship of proteins encoded by AD-associated genes was carried out in the string database by means of bioinformatics methods. Thus, it has been possible to identify the potential targets for the neuroprotective action of ultra-short peptides in AD. Let's talk about 4 short peptides, including pelon, epitalon, pinyalon, ivizogen. The drug cortexin was developed based on a polypeptide complex obtained from the extract of the cerebral cortex of young calves. The dietary supplement terlutan was developed based on a polypeptide complex obtained from the extract of whole cerebral cortex with white matter. The pineal peptide was synthesized based on analysis of amino acid composition of polypeptide complex from the extract of cerebral cortex of young calves. According to the different scientific data and bioinformatic analysis, it was established that the potential molecular mechanism of EGR peptide activity consists in the regulation of oxidative stress, the functional state of mitochondria and synaptic contacts, cell metabolism in AD. The drug Slavinorm and the dietary supplement Ventford were developed based on polypeptide complex obtained from the extract of the vessels of young calves. The Biogerontology peptide was synthesized based on the analysis of amino acid composition of polypeptide complex from the extract of the vessels of young calves. According to the different scientific data and bioinformatic analysis, it was established that potential molecular mechanisms of carrier-T peptide activity consist in the regulation of the functional state of blood-brain barrier, synaptic contacts, and neurogenesis in AD. The drug epithylamine and the dietary supplement endolutein were developed based on a polypeptide complex obtained from the extract of the epiphysis of young calves. The epithalone peptide was synthesized based on the analysis of amino acid composition of polypeptide complex from the extract of the epiphysis of young calves. According to the different scientific data and bioinformatic analysis, it was established that the potential molecular mechanisms of IgG peptide activity consist in the regulation of protein aggregation, neuroinflammation, oxidative stress and mitochondrial dysfunction, neurogenesis, as well as circadian rhythms impaired in patients with AD. The drug Timalin and the dietary supplement Vladonix were developed based on a polypeptide complex obtained from the extract of the thymus of young calves. The Biogerontology peptide was synthesized based on analysis of amino acid composition of polypeptide complex from the extract of the thymus of young calves. According to the different scientific data and bioinformatic analysis, it was established that the potential molecular mechanisms of carrier-peptide activity consists in the regulation of neuroinflammation, oxidative stress, and mitochondrial dysfunction. Thus, it can be assumed that short peptide may be the elements of the systemic regulation of neurodegenerative processes in AD by modulating the gene expression and protein synthesis involved in the development of pathogenetic links of Alzheimer's disease. Thus, in turn, it may underlie the cognitive enhancement in AD by short peptides. According to the several studies in the field of molecular modeling, as well as a deep analysis of scientific data, we suggest that a wide range of biological peptide activity is based on their potential ability to interact not only with DNA, but also with histone proteins, transcription factors, microRNA, and non-coding RNA, which are the molecules involved in the gene expression and protein synthesis. That's all I have to discuss in the present lecture. So, thank you for your attention.